Welcome to BrainXell's step-by-step guide to cell counting using a hemocytometer. Accurate cell counting is essential for research, ensuring consistency in experiments and applications. Before we begin, let's go over what you'll need. A cell suspension, a hemocytometer and cover slip, tri-pen blue stain, pipettes and tips, a microscope, protective gloves and a lab coat. Start by ensuring your cell suspension is evenly mixed. Clumped cells can lead to inaccurate counts, as automated counters may skip them, and manual counts may overestimate cell numbers. Next, mix equal parts of your cell suspension and 0.4% tri-pen blue stain. Now let's count some cells. Under the microscope at 10x magnification, focus on the counting grid. Visually inspect the 4 outer squares. Next, select the 2 most representative squares that are diagonally opposite each other. Follow a consistent counting rule. Include cells touching the top and left borders, but exclude those on the bottom and right. It's crucial to count both live and dead cells to determine if you have fewer cells overall, or if they have reduced viability. Let's calculate cell concentration using this formula. In the first square, count the number of live cells, and count the number of dead cells. Repeat that process for the second square. Add both live cell counts and multiply that by 10,000. This equals the number of viable cells. The final calculation of total live cell count divided by the total count of live and dead cells equals the percentage of live, viable cells. And if you are not using equal parts of cell suspension and tri-pen blue, you will need to account for a dilution factor. Be sure to reference the certificate of analysis for the expected live count of your specific cell lot, rather than the vial label. Happy counting! Please reach out to your BrainXell representative with any questions.